You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I have David Freedman, co-founder, president, COO of NanoView Bio. The website is nanoviewbio.com. We're going to be talking about characterizing exosomes and extracellular vesicles, EVs. Dave, thanks for coming.
My pleasure. Thanks for having me.
Yeah, for people that don't know, what are extracellular vesicles and exosomes?
Yeah, I like to use the analogy that I heard before of conveying them. They're like the Twitter of cells. So what they do is they are small, tiny messengers that cells secrete all the time. All cells secrete these. And they're ways for cells to transmit biological information from one cell to the other. So proteins, microRNA, other nucleic acids. And so they're critical in all parts of biology for intercellular communication. And this is only recently discovered, 5, 10 years ago. And so they used to think that these were cells just secreting trash or debris, but they're actually very important for all biological processes.
Huh. So people used to think cells were just throwing out the garbage, and that's what they were?
Yeah, exactly. For many years. I think they were discovered in the 50s or 60s, and so they kind of lay dormant for about 30 years. And then, and about 10 years ago, people really found that they were just critical parts of all biological processes. I think a lot of researchers are finding now that a lot of some of the science they discovered, 20 years ago, these exosomes or EVs could have been playing a role in those discoveries.
Yeah, this is huge. So what are the relative sizes of most people's, somatic cells, and what are the sizes of the exosomes or EVs they put out?
Yeah, so I think the number most common is for regular cells, it's about 10 microns, 10 micrometers in diameter, something like a fifth the size of a human hair. And exosomes it's sort of hotly debated, but I think generally speaking, they're somewhere between 30 and 50 to 200 nanometers in size. So I think by volume, they're about 1 million times smaller than, one exosome is about 1 million times smaller than a single cell.
Oh, wow. They must be very hard to find, I guess, in a lot of analyses. I mean, they're in there, but are they even viewable? Like, how would you view them? Can they be viewed?
Yeah, well, that's a great question. It's one we like to answer over at NanoView because we have a great way to visualize and characterize these exosomes. But generally speaking, they're much smaller than the wavelength of light, so it's difficult to use a traditional microscopy technique. So there are some technologies that are low throughput, like atomic force microscopy or transmission electron microscopy, or TEM. Those are very low throughput, but they're useful for getting really ground truth information about what's going on. But more commonly used for understanding your EV, your extracellular vesicle population, is a technique called nanoparticle tracking analysis. And what this does is it can tell you if you have nanoparticles circulating in your fluid and can count and size them. But it is lacking in really being able to easily measure any biospecificity or something related to those particles' biological prevalence.
How do people even know that these EVs are there if they're so small? Like, has anyone been able to observe them in vivo, or is it completely impossible? And I mean, how were they found originally?
Yeah, so I think they were found originally, or in the very early days, people thought they were trash in these transmission electron microscopy images. And then there was a landmark study where it showed that they purified them, so they run a very laborious purification process to try to get these EVs from typically cell culture or in vivo from a blood sample. and then they take those EVs and they put them into another system, another cell culture media, and they should not have expressed a certain microRNA profile. And what ended up happening is by putting in these EVs, they saw that the cells uptook them and then started expressing these profiles. So that was sort of evidence that this was happening. And so they have verified using transmission TEM, transmission electron microscopy. They can purify these samples, so it's a very laborious, about 8-hour prep where you can get a very purified sample, and then they put them down on the imaging grid, and then they were able to see these tiny vesicles.
Do you think that these vesicles are responsible for most of cellular communication, or just a small fraction?
I think that's a great question. I think it's one that we still don't know the answer to yet, but my personal belief is that they're probably a critical part of that intercellular communication. I mean, I think there's other things circulating in biofluids, but I think you'll find in the coming years that because they're able to package up these molecules, they can protect them in the bloodstream for longer, and so potentially they have a longer and then a more positive uptake in downstream cells.
So is there an organelle inside the cell that packages these and sends them out? And is there one that takes them into the cell and opens them up and, does something with the contents?
Yeah, so I think those are open questions. I think there's some theories about that, but I wouldn't want to speculate on them today.
Has anyone ever, again, so when you observe these vesicles, you have to purify them and just view them, I guess, in isolation, and they're not active, they're dead, or?
Well, so they're not active. Yeah, no, that's a good question. So I actually didn't fully answer your previous question, so let me answer both of them. So they're not active biological processes, so they'll contain similar machinery, but not the active machinery to generate things. So they will package up, say, some proteins or microRNA, but they're not actively creating things. And to your earlier question, there are techniques that people have used, typically fluorescent microscopy techniques to look at stained cells, and they have stained and genetically modified these some EVs to be able to enhance a fluorescent molecule, and then they'll use that to see that the cells uptake them preferentially, and certain types of cells will uptake specific EVs. So they have used that as additional evidence to demonstrate the possibility of EVs being involved in all these biological processes.
Well, has anyone tried to put a given cell in the presence of, thousands or millions of EVs that the cell would take up, let it take them up, and stop the process at some point and observe the cell internally and see if you can still find evidence of them intact in the cytoplasm of the cell or in various stages of disassembly?
So I don't believe people have, I'm not familiar with them looking at various parts of the cell, but they've definitely run studies similar to what you've described. And what they've seen is a longer lasting effect. So, for example, the cells don't necessarily stop showing that uptake immediately, although the effects do wane off after a period of time. I can't remember if it was multiple days or weeks, but they have run studies very similar to that, which are quite interesting.
So it's still like black box. It's like cell black box spits out these vesicles. When they go into another cell, we don't know what happens to them. We don't know how they're made or packaged or opened up or utilized or any of that yet, right?
Yeah, so I've heard this analogy. It's like immunology was 30 years ago. So yeah, we're still there's a ton to learn about these EVs and exosomes and how they are made and how they're preferentially packaging up cargo, and how they then preferentially go to cells downstream from them. It's all really exciting stuff that is, I think, on the forefront of technology and science.
And how many are put out by cells and how many are uptaken? I know it depends on conditions, but is there, do cells seem to put out just a few or tons of them? Or
there's varying reports in the literature. I've heard numbers as small as tens for some cells that are not under a great deal of stress, upwards of millions for cancer cells that are just spewing them out. So probably somewhere in the middle is where you might see it land in the future. But yeah, I think depends on the cell type, its environment, and things like that. So it varies dramatically.
What about the vesicles themselves? Like, I guess that's probably the first place to characterize them. Like, what's inside them? How much energy does it apparently seem to take the cell to prepare them? And then, how are they used? What are they communicating?
Yeah, I mean, I think that's a lot of what we're trying to do here at NanoView is understand some of these questions. So, we've developed a platform that lets us look at the outside and inside of the exosomes in a high throughput way. And so some of the questions we're trying to answer are around that. So, for example, we see common sets of markers. This is reported in the literature, the tetraspanins that are on a number of exosomes. And, one question, for example, there's these three markers is do exosomes contain all three? Do they only contain one at a time? And what concentration? And so from our early studies, we're seeing it, all types, right? We see some samples, some cells that are secreting exosomes that contain all three of these tetraspanins. Other cells that we're seeing, other cell types are secreting, only one of these in abundance. So right now it's still early. I wouldn't draw any specific conclusions, but a lot of exciting things happening.
Do they appear to have internal structure, or what's inside the exosomes?
So again, I like that analogy of the mini cell or the Twitter cells because essentially they contained a bilayer, lipid bilayer that kind of protects them, and then inside it can really be anything. Particularly, we're seeing proteins on the membrane or inside the EV or exosome. And one of the most common things people study as well is microRNA. We've seen also studies showing that potentially they also package up DNA as well, fragments of DNA. So it's really like I like the example where it really just is this protector, this lipid bilayer, and inside it will contain something that seems to be preferentially packaged from the parent cell, but not universal across the exosomes.
Is there any activity going on an exosome, or is it just passive and floating around and then binding? Is it just a container, is what I'm saying?
I think of it more as the latter. It's really just a container. What causes it to do preferential activity is the markers on the surface. So they'll often express different surface markers. Again, that's where those tetraspanins that we were talking about earlier are commonly shown to be on most exosomes. Then there'll be other surface markers, for example, something specifically targeting a cell from a cancer cell. So there's been papers showing integrins will be on the surface and that these will seed metastatic sites. So a lot of things can really be on there, but I think of it more of a passive carrier as opposed to kind of an active process.
Do they only appear to bind to cells, or do they? They float around. I guess they go through interstitial fluid. They go through the bloodstream. There's so many questions. Are there places in the body that they preferentially go? Can they go anywhere?
They seem to be implicated in all areas. Some studies are showing there's a lot of excitement about looking at them for neurodegenerative disease because they're believed to cross the blood-brain barrier. And then there's other studies that, for example, have shown that pancreatic cells will secrete exosomes that will specifically target the lung and other organs. And so that's potentially why metastatic sites from specific cancers will see different specific metastatic sites, remote sites. So I don't know exactly why that's happening, but there are studies showing that is seems to be the case.
Well, for instance, in cancer, is it believed that the exosomes itself are being absorbed by normal cells, turning them cancerous, or is it? I don't know. Is there any scouting and reporting back mechanism, or is it once the exosomes are out of the cell, they're not coming back? Is it like a one-way communication?
Yeah, that's a good question. Yeah, I think I've seen mostly the latter, where that they're not scouting and reporting, but they're actually going out and kind of more like scouts only, or really their artillery just being deployed remotely. I guess maybe covert action, like Navy SEALs, is more appropriate because a small number of them will then go there and kind of set up shop to make them appropriate for cancer cells to then come off and metastasize. So there was a landmark study, I think it was almost 2 years ago now, showing that these exosomes create these preferential niche sites for cancers to then cancer cells to start growing. So they'll seed that site is kind of the studies that I've most recently read.
So that means what? They'll bind to existing cells in a given area, and then when cancer cells migrate around the body, they'll see the markers, and then they'll stay there and attach, and that'll start a new site for a metastasis, for instance?
Yeah, more or less. I think that's a pretty good analysis of what the studies I've read.
But there's no, I guess to put it frankly, the exosomes are not alive. There's no complicated, sophisticated action that they undertake. There's no, what if they form something like a biofilm, essentially, or if they were able to have some kind of action on their own besides just being containers?
Well, they can through the delivery of that cargo. So some of that cargo, so that cancer analogy, I think, is an example of that. So they'll basically lower the barrier, the activation energy for a cancer cell to then metastasize somewhere else. So they're delivering cargo. Now, what that cargo is, don't really know. I think that's one of the things that we're trying to study here at NanoView. But that potentially is delivery of some mechanism. So typically it's microRNA or proteins, and so then they'll downregulate or upregulate different biological functions in the delivery cell.
It's just amazing, though, because I know of no other, I don't really know of other.
I think viruses are a good analogy. They're not necessarily replicating what they're delivering, but I think a virus, we did a lot of work with viruses when we originally developed this company, and I think we pivoted in the exome space because for us it was very similar from a technology development perspective. We had a platform that can look at biological nanoparticles based on their surface markers. So, of course, viruses use their host cells' machinery to then replicate and infect others. I don't like to use that analogy with exosomes, but they have surface markers and contain cargo very much like a virus. And so they're doing something like a virus in that sense. But it's not an analogy we like to use, but I think it helps, at least for me, understand kind of the biological mechanisms and that they're delivering something and then causing the cell to do something different.
So do you think they're also, again, attaching or entering normal cells and turning them into cancer cells?
I have not seen studies that say that, but that is possible. I think that's a hard question to answer about whether they're causing the genetic mutation, but they can typically upregulate or downregulate different activities.
Well, by doing so, I guess it's possible they could turn on oncogenes, possibly, and change a cell into a cancer cell, maybe.
Maybe, yeah. I don't want to. I haven't read enough studies to be confident to state that, but that seems like a reasonable hypothesis that should be tested.
Well, have people tried to culture exosomes? Have they taken a cell population, harvested their exosomes, cultured them to large amounts, and then injected them back into, let's say, a mouse to see what kind of effect it has on the mouse? So
they have, but I'm not familiar with the cancer studies. I'm much more familiar with the studies where they're using them for regenerative medicine. So there's a lot of companies that are using exosomes. So they typically were stem cell therapy companies. And what they found is that they can take the exosomes instead of the stem cells and have the same kind of therapeutic effect where if they inject a large number of these exosomes into a diseased person or animal, they can recover that function. For example, there's ocular regeneration or cardiac regeneration from these exosomes. So those studies seem to implicate that what you describe should be happening as well. I'm just not as familiar with those studies.
Right. I just think it's so fascinating. I know I'm spewing out questions like cells spew out exosomes, but
no,
there's so many.
It's super exciting. I think that's what we got so excited about as well. It's just the space is exploding. There's so much to be learned, and so I think that's what we got so excited about here at NanoView.
So what's your focus? What area of this whole new field essentially are you focused on?
So we're focused on helping other people develop and answer these questions. So I think when we started this, what we learned is it was very, very difficult to, especially without that laborious purification step, be able to understand what's going on with these exosomes. So traditional workflow is to do ultracentrifugation to get a really purified sample that's pretty low throughput. Then they'll take that and then they'll run it through NTA to make sure that they actually have nanoparticles. And then they'll run kind of either a Western blot or some other assay to understand that there's some biological properties that they're looking for. And so what we've done is collapse all that into a single step, and that allows us to really understand what's going on in a high throughput way. So we can run many samples here, and we can do it without doing that purification step. And then we collapse and get the nanoparticle size and count along with its biological activity. So I think we're helping answer companies that are developing liquid biopsies or diagnostics using these exosomes, as well as companies developing and researchers that are developing new therapeutics, because these are natural biocarriers and they can load them up with therapeutic cargo and deliver them to specific cells through those markers. There's potentially a huge opportunity for new classes of therapeutics. So all those things combined are what get us really excited.
How many, for a given cell, how many different types of exosomes does it appear to create? Are there many different flavors and, in response to different stresses? Does it make completely different exosomes, or does it stay within a narrow range of production?
So I think that's a really interesting question. I think the assumption is that there's different exosomes being secreted under different stress types, but coming up with kind of a unique fingerprint for exosomes is something that we're actively working on. And so I think the question is what defines a specific class of them. So that I think is still being answered, and we're working to help the community understand this, but is 3 markers defining it? Is it 10 markers on a single exosome? So what we're seeing is that there's a whole potpourri of different types of exosomes coming from just for example, a single cell culture. But yeah, I think understanding if these are similar, the size difference making them different, I think those are questions that we're helping answer.
Huh. And I wonder how our cells in our body filter. So I would think that, in our body, there's just crazy amounts of exosomes everywhere, which would create a ton of noise. Let's say I'm a heart cell. I would only care, I'm sure, about certain exosomes and most not. How do I filter them? How do I get through the noise and still do my job and, not be overwhelmed by it?
Yeah, I think that comes down to what's on the surface of those EVs and then what cells will kind of accept them for delivery. I think those are kind of really interesting questions, and so people are looking at PDL1 and PDL or PD1 for immuno-oncology and understanding why some exosomes are upregulating or downregulating activity. It's a really interesting question. Still think very early though but you are right that there's a lot of exosomes circulating in everyone's body, and so understanding why some are going somewhere and some are going another place, it's a really interesting question.
Well, how would even a cell know, okay, I'm going to make an exosome that's going to target these cell types? How would they, it's just weird, the whole question. How would they even know what to make? Has anyone done this? Has anyone taken, for instance, all the cell types in the human body, characterized their outer layers, and looked for differences? Because then maybe perhaps you can create a map of, okay, for a given exosome, which cell types could this interact with? I wonder if you could do something like that to create this crazy map of the possible interactions and stuff.
Yeah, no, I think that's a really interesting question. I think I'm not aware of work like that done in the field, and I think it is incredibly difficult just because of the traditional techniques, low throughput, and the ability to accurately characterize all these samples. So maybe with better technology like what we're developing at NanoView, that can help answer that question. I know that they have looked, for example, at different cell types in culture to understand how many exosomes they can produce. And so one kind of common cell type used in the field now is mesenchymal stem cells, because they secrete more exosomes than most other cells. So at least in culture. So those, I see those commonly referred to in the field, and I think it's because an earlier study kind of found that they secrete more exosomes than other cells, which makes sense. They're early cells, so maybe they're doing more scouting work or things like that in terms of figuring out what the kind of cell they should be. I'm not really sure why it is, but I think it's really interesting that that was found.
I mean, even within a given organ, let's say again, the heart, how do the cells know how to coordinate within that organ to achieve its function? So maybe there's a class of exosomes that are just cell type specific, so they know that this is targeted for other heart cells like me, and then there's a class that are targeted for, not me, other organs, other parts of the body, other cell types. Maybe there's that hierarchy of exosomes, different types.
I think there are some of those. I'm not familiar with them. I know that we're working to develop things like a cardiac panel and other things that are specific to different organs. So that is the case. I'm just not familiar with which markers those would be. And to your kind of biological question, I mean, why did they package those things specifically? I think those are all really interesting questions.
Yeah. Well, here's something even crazier. So our microbiome. Does our microbiome create exosomes that interact with our cells and vice versa? And is there communication going on that way?
Absolutely. I mean, that could be happening. I know I've read a number of different microbiome-created exosomes, and what interaction is happening between the host and the microbiome. Those are really interesting questions that I think people are starting to study in the literature. And how they're affecting it, those are all kind of really interesting things.
Yeah. I'm also wondering about, conception too, like sperm and egg. I wonder if the egg is putting out all kinds of exosomes that are telling the sperm something, and the sperm are doing something, and then maybe one puts out preferential exosomes that allow it to access when others don't, and if there's any evaluation and vetting there before like fertilization occurs, instead of just like mechanically trying to burrow into the egg.
Yeah, it'd be interesting. I know we've looked at reproductive fluids, and we do see exosomes in there, so there must be. I don't know if they're coming from the sperm or the egg. I'm not sure I want to make that claim, but I do know that there are exosomes in those fluids, so something is going on, sure.
Jeez, yeah, this is a whole new field. It's crazy.
Yeah, it's really exciting. I mean, like you said, the immunology was, we know so much more now than we did 30, 40 years ago. I feel like the exome space is, you have all these really exciting questions. I think people are really trying to answer them. And that's where we come in and we help them. I think we're helping answer those questions for researchers. And I think we're going to look back in 10 years and we'll know so much more than we do today. So it is really exciting. And I think what we'll see is a lot of diagnostics and therapeutics that come out from all this research.
Well, if you ever run out of questions, let me know. I've got like 8 million more.
Sure, of course. Yeah.
So all right, so there's millions of things that could be done. What are some of the focuses that you have? What are some, 2 to 5 year projects that you're working on that you think will come to fruition?
Yeah. So in the exosome space, I think the largest interest is around oncology. So I think there's obviously a lot of research happening in that space and a lot of exciting findings, and I think that's probably one of the most advanced areas. And so there is, for example, a CLIA test on the market for prostate cancer, and so using exosomes and cell-free DNA. So I think that's kind of at the front edge of what's being used for exosomes. So oncology, immuno-oncology space. At NanoView, we're looking at developing immuno-oncology panels in collaboration with our partners that we hope become kind of a standard offering for all customers. And then the other kind of hot topic area is neurodegenerative. We sort of touched on that briefly. Because there's a belief that they cross the blood-brain barrier, there's the thought that potentially diagnostics or therapeutics for neurodegenerative diseases could be developed without being invasive and getting into the brain. So that area is exploding, and again, we're excited about the developments we're doing here at NanoView for that. The other 2 areas that you see a lot of research papers coming out is cardiac. So that's another area that people are doing active research in, as well as reproductive biology, I guess, and looking at in vitro fertilization or diseases such as I'm blanking on it. We'll pretend I didn't say that. So women's reproductive health yeah, that area is also a lot of diagnostics, non-invasive diagnostics are looking to be developed there. And then, as I mentioned previously, I'm really excited about the therapeutic space because I think the ability to package up therapeutic cargo with specific targeting markers is kind of a holy grail for therapeutic development. And so if people can, and these are natural biological molecules, so they can evade the immune system longer because they present some of the same non-pathogenic markers on their surface. So the potential for better therapeutics, I think, is huge. So that's one of the space that we would like to work with our partners to help advance that field as well.
Have people observed that bacteria put out their own types of exosomes or viruses?
Absolutely. Yeah, viruses not so much, but definitely bacteria. I've seen a number of posters at different conferences where people talk about the potential for using them. There's been some that I thought were a little out there. I'm not remembering the titles exactly, but that they, I think, related to like malaria and how the exosomes can help, I think, increase the infection because the bacteria in the mosquito are helping up to reduce the barrier for the infection. So there's things like that. So bacteria are definitely one of the interesting areas to study, and obviously it's a nice system to also study under.
And I guess if you were to create a protocol, let's say something that gets injected into the bloodstream and it sucks up a particular type of exosome and binds it, that may be a way to resist infection, way to stop cancer, metastasis, et cetera.
Yeah, I mean, I think that there's potential. Well, I think maybe injecting, specific classes of exosomes could, with different specific makeups, both on their outside and inside, their cargo and their unique identifier on the outside, that could have the potential for the ability for diseases to replicate.
Yeah, I mean, to stop, again, some kind of bacteria or from infecting us, again, if it puts out a certain type of exosome, if we were to make an exosome sponge, for lack of a better word, that we can inject, that would suck them all up so that the bacteria couldn't take hold or make it much harder to do its job to attack us. For just an idea.
Yeah, you mean like some exosome decoys or something like that?
Yeah, or again, like a sponge to suck them up, to take them out of the fluids.
Yeah, it's an interesting idea.
Well, it sounds like there's a ridiculous amount of science to be done still.
I think there's still a lot of work. I mean, it's a lot. Obviously, it's really exciting because, I think I just read an article that came out today about not using, they were putting, I think, exosomes on skins to prevent wrinkles. So there's a lot of work being done in the exosome space. I think cosmetics, there's been a number of different companies started up to use exosomes as a topical cosmetic, and they seem to be selling products. So, I mean, I don't know about the science. I haven't really investigated these things, but I think it's interesting that people are looking towards them as a alternative solution.
Yeah. So, all right. So, some resources for listeners. How can they find out more about NanoView Bio and get in contact?
Yeah. So, I'd recommend the best place for information is nanoviewbio.com. That's where we have most of our public information. You can also check us out at Twitter at NanoView Bio, and look for us on LinkedIn as well.
And I haven't asked this before, but any keywords that come to mind? You said you read this study, you saw this, you saw that. What keywords do you use, or can listeners use, to search and learn more about this space in general? I guess exosomes, extracellular vesicles, anything else?
Yeah, exosomes, extracellular vesicles are probably the most common terms. To really learn about the community and the field, I recommend the two international sort of central bodies. So the International Society for Extracellular Vesicles, that will be in that annual conference will be in Philadelphia next year. And then also the Journal for the Society for Extracellular Vesicles, the JSEV. That is a open access journal that is focused on the exosome community. So those are, I think, excellent resources that you can check out, and those are by the kind of global scientific community focused on advancing exosome development.
Okay, excellent. Well, this has been a great call. I'm excited.
Wonderful.
You're excited.
Awesome,
So,
Richard.
I appreciate you coming.
Oh, my pleasure. Thanks a lot, Richard. This is fun.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.